later stage of cell culture requires more passages. Some of the problems that are not the problem at the early stage will become major problems down the road. We have seen that from our licensees, larger companies, pharmaceutical companies that license our iPSCs, and comparing to other iPSCs in the field, they will see the difference. So a line, just because it's been around and it's available to the field, does not necessarily mean it's a good line. So sometimes the companies need to actually spend more time or even more money to make sure the lines work in different stages. They can do design studies to see the cell stability in terms of their morphology, their post-thaw thawing viability, their telomere length. We actually have seen from our clients using third-party analysis to see the distribution of telomere length and passage-to-passage consistency. And along the line with that is the genome stability in general. So we have seen first-hand data. The iPSC line quality itself can dictate all of these down the road important attributes of iPSC cells.